Tropical and Public Health Institute here in Basel. Dr. Brun has pointed out the key role of DNDi, the Drugs for Neglected Diseases Initiative in Geneva, in making new drugs for parasites. I would like to start by reminding us of their motto, best science for the most neglected, which is actually more than a motto. I think it's a strong statement not to make any compromises in terms of quality of research and quality of development for drugs for neglected diseases. That shows their very strong commitment. But of course, such a commitment comes at a price. We learned 2 days ago from Patrick Hunziker's talk that the costs for development of a new drug are estimated to be more than $2 billion, I think he said. Of course, that incorporates the cost of all the failures. It's not only the cost of one product. And basically, these are sums that may be returned by a blockbuster drug within a year, if you take a cholesterol-lowering drug. But obviously, it's absolutely impossible for drugs for neglected diseases to ever make such a return. So clearly, we need funding schemes, funding ideas. New incentives are needed to get people into drug discovery, particularly into development of the discovered drugs. And there we have, on the one hand, what you all know, these sort of push mechanisms. That would be DNDi or MMV or what they're all called. So we heard about that yesterday too, how much government invests in research, driving research. On the other hand, I think it's very interesting also to look at the pull mechanisms. So we have these are the product development partnerships. I think is a very good example here of push mechanisms. The pull mechanisms try to create incentives at the end. And here I would like to quickly introduce this FDA priority voucher system. So that's I think a very creative idea by the Food and Drug Administration to create incentives. It's actually not that new. It was signed by President Bush in 2007. And it states that if you develop a new chemical entity for any of these neglected diseases, later also rare pediatric diseases were added there, and get it approved by the FDA, you can get a priority review voucher, a voucher that entitles you to have another file handled within 6 months than within the normally 18 months. And you can use that for another drug, but you can also sell it to a company and they can use the voucher. So here Bill Gates, a year afterwards, immediately stated that if you really have a blockbuster like a cholesterol-lowering drug, it could go on the market a year earlier, and thus the priority review may be worth hundreds of millions of dollars. So far, there have been 4 priority vouchers given for drugs for neglected diseases. There's more vouchers actually were given for rare pediatric diseases. The last one, interestingly, was for a vaccine, cholera vaccine, last year. The first one was given to Novartis for Coartem. But somewhat unfortunately, Novartis used that voucher for one of their own products, which eventually failed. It didn't get registered by the FDA. So we didn't really have the $1 million value. And the real breakthrough came somewhat later. There was a big head-to-head race for the next generation of cholesterol-lowering agents. So these are Praluent by Sanofi, Repatha by Amgen, and these were the first generation of drugs that don't inhibit the synthesis of cholesterol. And here, interestingly, Amgen was ahead. They got their product registered by the EMA first. They bet Sanofi. But with the FDA, Sanofi put a fast-track voucher on the table, and they actually got Praluent approved in July 2015, and they bet Amgen by a month. Which you can imagine for something like the new generation of cholesterol reagents to be the first on the US market is extremely valuable. And it's interesting to note that, so that happened in July 2015, and then there was a record-selling fast-track voucher sale only in August. A month later, one of the fast-track vouchers was sold for $350 million US dollars. So that is still today the record price ever achieved for such a voucher. So I think you agree with me that there is funding, as Reto already alluded to. So we have, on the one hand, the pull, but also these interesting push initiatives. What we need now is new ideas also, maybe new approaches to attack parasites. And there's one thing I would like to share with you. If we think about all these parasites, we heard about the helminth, the malaria. To me, a big difference between the parasites to, let's say, cancer cells or bacteria is that they have these cycles, which are often quite complicated, which we have to learn for university, which nobody knows better than our next speaker. So this, for instance, is a very simple cycle. That's Haemonchus, gastrointestinal nematode of sheep. Actually, it's a nematode where drug resistance is a big problem. So we have adults that live in the abomasum of the sheep. The eggs are shed. They develop on the grass and the pasture. Finally, we have this larva 3 infective stage, which is kind of a sleeping stage. It just waits there. It can wait for months on the grass until it's eventually eaten by a sheep, where it will develop into larvae 4 and into adults. Now, Ralph Sommer and colleagues of the Max Planck Institute for Evolutionary Biology in Tübingen, they found out that these processes, the sleeping larva, is actually related to the dower larvae of C. elegans, and they found that they can, by using dower-repressing pheromones, wake up these larvae, and they develop into larvae 4 before they're in the sheep. So they develop in the wrong place, and they will die. The larvae 4 can only survive in the sheep. So basically the idea is to use this cycle and trick the stage into thinking it's already in the sheep, and it will develop at the wrong time or in the wrong place. And if the concept is applied to maybe a more complicated cycle, that's now the cycle of Trypanosome brucei, the sleeping sickness parasite. The idea was exactly the same, to trick these stages that multiply in our blood, in the mammalian blood at 37 degrees, to think that they're already in the fly and develop to such stages here, which are in the midgut of the fly, and they would be doomed. They could not survive then in the fly. And that was exactly the idea by Reto Brun and Isabelle Roditi of the University of Bern. And Isabelle made this reporter strain, so that's a genetically modified T. brucei strain. We have here the procycling genes encode the surface protein of the fly-formed trypanosomes. And this locus is silent, it's not expressed in the bloodstream stages, and they inserted the reporter gene here that allows for a fluorescence screen and screen for small molecules that would cause expression of that locus in the bloodstream stage. Tanja Wenzler in the lab actually carried out screens. She, by now, I think, screened close to 300,000 of molecules. I just show you one example here of a hit. So we have a molecule that's all at 37 degrees in trypanosomes that induces expression of that procyclin locus, and at the same time you see that's killing the trypanosome. So it's an untimely, uncontrolled differentiation of trypanosomes. So I wanted to show you this as a proof of concept that we can actually attack parasites by messing with their cycles, by tricking them, by using chemical triggers that makes them differentiate at the wrong time. So we have new funding opportunities. We have new ideas. That's just one of them. I'm sure we will hear about more new ideas later. So what we actually need now, of course, is the new medicines. Thank you very much. time for a question.
Is it unavoidable going to the pharma for developing medicine for a neglected disease? Because sometimes I have the feeling that because they have to invest so much money in the development, that then either this medicine cannot be sold where it has to be used, or it will be sold at such a price that it cannot be properly used. So I don't know if there are other ways to develop a product for these diseases.
Yes, me too. I wouldn't know of an alternative. I think there's some steps already in preclinical development, like the full tox, all the formulation. That's, to me, clearly pharma know-how that's needed to make a product. And then you need professional developers. It reminds me a bit about the talk yesterday. Who do you want to build your house? Is it the cement expert or the builder? So I think the builders, the developers are with pharma. So. But there may be, yeah, maybe there's alternative ideas to build up new schemes. Yeah.
Sorry, I came in late, but that has always been my problem. And the only way you can actually develop a product for neglected diseases is through public-private partnership. There's no pharmaceutical industry who will invest their money to come up with a drug of poverty because they won't be able to pay their money back. So yeah, the basic research is fine, but when it comes to product development, it's very expensive and you need a PPP. Thanks.
Yes, so that's exactly, I think, I would say the good situation we have today, that we have these means, thanks to the Bill and Melinda Gates Foundations and also to many other. We also learned about philanthropists yesterday, so right now I think the situation looks much better than, let's say, in the 90s before we had all these PDPs.